Okay, great. Our next speaker in this session will be Pujitha Macha. So Pujitha is a master's student in the Department of Computer Science at the University of Saskatchewan and a research assistant in PERC, working under the supervision of Dr. Mark Aramian and Dr. Leon Koshin. Pujitha recently received the MSc Citizenship Award from the Department of Computer Science for actively contributing to various activities and events on campus. So she's definitely an awesome contributor to our lab and to our department. And she's also worked as a software engineer at Bosch Global Software Technologies in India before moving to Canada. And today, Pujitha will present on a kind of overview of the work on high-throughput automated phenotyping pipelines for 2D and 3D plant root images. So we're trying to highlight here a nice cross-section between flagship 3 on data analysis and flagship 2 on the root imaging work. So I'll invite Pujitha now. Thank you.
Hey everyone, I'm Pujitha Macha, and I'm here to talk about high-throughput automated phenotyping pipelines for 2D and 3D plant root images. Our team at the University of Saskatchewan doing digital analysis has been working on some really interesting projects with Global Institute for Food Security. Here's some of the cool stuff we've been working on. So I'll be giving a high-level view of these 3 projects. In 2D image space, we'll look into image segmentation of plant root images. And then followed by, we'll look into semantic segmentation and abstract representation and how this step is helpful in computation of more complex phenotypes. As for 3D image space, we'll look into in-painting of occluded roots using generative adversarial networks. Kyle Sidenthal and his team worked on iterative image segmentation of plant roots for high-throughput phenotyping. So this is a joint work of UFS with Global Institute of Food Security. So here's how a 2D plant root phenotyping pipeline looks like. It takes a set of 2D RGB plant root images as input, and it outputs plant root phenotypes. We'll now look into the segmentation module. This is a high-level view of iterative neural networks that we use for segmentation. It's iterative because if you see in the image here, it has a set of ViewNets that are connected with the help of skip connections. And the output of one network acts as an input to the next network, and the output of the last network as the final segmentation mask. The motivation behind using this iterative networks is that if you want to be successful in segmenting thin road networks and satellite road images. So to train and test the model, we require thin and complex root networks. So for annotation, we've used in-house developed annotation tool that is friendly ground truth annotation tool. The figure here provides a screenshot of an in-progress annotation on a patch. So this friendly ground truth annotation tool takes a large image as input, and it splits it into non-overlapping patches. We can work on one patch at a time without having to manage this entire image. Moving on to data sets. We use 6 different data sets to train and test the model. So out of which we have 2 sets of cucumber data sets and 2 sets of soybean and 1 wheat data set. These 5 data sets are provided by Dr. Kochian's group at the Global Institute for Food Security. We also used canola images from Agriculture and Agri-Food Canada. The data is split into training, testing, and 2 validation sets. One of the cucumber data sets is excluded from training, testing, and the first validation set. This is done to later analyze as to how well the model generalizes to new data sets. So here are some of the sample images that I just mentioned in the earlier slide. So if you observe here, these images have varied root structures, and it also has a measuring scale, which is a non-root object. Moving on to model predictions. So these iterative networks take image A as input, and it outputs a segmentation mask. Yeah, so image B is the mask predicted by iterative neural networks. You can see that the model did a really good job in extracting plant root from the background. Another thing I'd like to highlight is that it was able to ignore these non-root objects like measuring scale. So this helps in high throughput applications where we don't need to have the explicit auto-cropping step or human intervention to crop images. take a closer look at the segmentation results. For image A, simpler root structures, in this image, it has a die-score of about 0.96. As for complex root systems with greatly overlapping root structures like an image B, this image has a die-score of about 0.87. Okay, moving on to semantic segmentation and abstract representation. Dr. Mark Iramian and I have been working on this project. Most of whatever I'm going to say under this project is a work in progress. So this is a joint work of UFS with Global Institute of Food Security. So revisiting the pipeline from the earlier slide, Kyle Siegenthal's iterative networks, it solves the segmentation problem, and we'll now look at a semantic segmentation and abstract representation. So semantic segmentation, it not just detects root pixels, but it also classifies them as primary or lateral root pixels. So this step provides critical information for constructing an abstract representation of root system architecture. There are two ways to go about this. One is pixel-based semantic segmentation. Another one is graph-based semantic segmentation. So pixel-based semantic segmentation model will try and classify each pixel as either primary or lateral root pixel. But plant roots, they essentially have this tree-like structure where we can consider branching points as nodes or root segments as edges. So we can leverage the structural information and perform a graph-based semantic segmentation, where we'll try and classify each edge as either primary or lateral root edge rather than doing it pixel-wise. So image A here is a label plant root image, and image B is the skeleton version of it. So label plant root image, it helps in extraction of standard phenotypes like total root length and total root width. The reason we don't just want to stop at image A, but also extract skeleton is that it helps in computation of more complex phenotypes like branching angles. So to support this graph-based semantic segmentation, we've been working on graph-based encoding technique that encodes this node and edge information into a tensor, and we can use these tensors to train neural networks. Moving on to 3D image space. Hao Song and his team, they've been working on in-painting of occluded roots using generative adversarial networks. This is a joint work of UFS with Global Institute of Food Security and NRC. So I'd like to introduce you all to the data acquisition process first. So to maintain the 3D structure of roots, we're using this mesh tower. So if you see an image A here, a person is transferring seed or root to mesh tower. Once this entire setup is ready, we'll be transferring that setup to a solution tank for root growth. So these solution tanks are temperature controlled. Once the root images are ready to be captured, we'll be capturing a set of 2D RGB images by rotating the mesh tower over 360 degrees. Say for a plant, we would collect about 100 high-resolution 2D RGB images with the help of the 3D imaging system shown in MHC. So we'll be later stitching these 2D images into a 3D model. So image A here is a sample image that is captured using the 3D imaging system. You can observe that due to the mesh tower structure, parts of the roots are occluded, but it was necessary for us to retain the mesh tower because that helps in maintaining the overall structure of roots. But we'll be using a segmentation method to extract the roots, and you can observe that extraction of segmentation mask resulted in disconnected roots due to the mesh tower. We'll be using generative adversarial networks to reconstruct the missing parts of the root. So generative adversarial networks, they are generally used to generate images. So as for the model prediction goes, image A is for a simple plant root image. Firstly, the red pixels are the pixels predicted by generative adversarial networks. For a simple plant root image, it has an accuracy of about 95%, but for a complex root system like an image B here, it has an accuracy of about 85, but it's still doing a really good job in reconstructing those pixels back. So this is how a 3D pipeline looks like. So this 3D pipeline takes a set of 2D RGB images as input, and Kyle Siegenthal's iterative networks, they solve the segmentation problem. Hao Song's work is focused around this root gaps in painting step, where we'll try and reconstruct the missing pixels back together with the help of generative adversarial networks. And once we have these reconstructed segmentation masks ready, we'll be stitching them into a 3D model. And we can use this 3D model to extract traits like root fresh weight and root volume. Or we could further process it to extract abstract representation that helps in computation of more complex phenotypes. To sum it all up, we just looked into iterative networks for segmentation, and we also looked into this additional step that is semantic segmentation and abstract representation, and how that is useful for extraction of more complex phenotypes than the standard ones. In 3D image space, we looked into impending of occluded rules using generative adversarial networks. Our team at the University of Saskatchewan is trying to make changes to both 2D and 3D pipelines to not just make each step more accurate, but also to enable high throughput applications. Thank you for listening.